Good morning, everyone. I'm John Christian Giagustin, your reporter for today. We are going to tackle about cell motility. Before we proceed to our main discussion, first let us know the terms that we will encounter throughout our lesson. First, the motility. In biology, motility is the ability of organisms and fluid to move or get around. Cilia. Cilia are microtubule-based hair-like organelles that extend from the surface of almost all cell types of the human body. Flagellum. It is the hair-like structure that acts as primarily as an organelle of locomotion in the cells of many living organisms. Cytoskeleton. It is the structure that helps cells maintain their shape and internal organization, and it also provides mechanical support that enables cells to carry out essential functions like division and movement.
What is cell motility? Cellular motility is the spontaneous movement of a cell from one location to another by consumption of energy. The term encompasses several types of motion, including swimming, crawling, gliding, and swarming. It is a highly dynamic phenomenon that is essential to a variety of biological processes such as the development of an organism or morphogenesis, wound healing, cancer metastasis, and immune response. So our body relies on our cells and tissues to function properly in order to stay healthy. But it also relies on these cells and tissues to be in the right place at the right time. So cell motility, it helps ensure that our cells get to where they're supposed to be. That is especially important in developing tissues. And these cells develop into mature tissue, then migrate to wherever they're supposed to go. So for example, we have a wound in our skin. So without cell motility, our skin would not be able to regenerate itself properly, which would have far-reaching effects for your health. And the same concept applies to other tissues. Mature cells that can't migrate to the right place in your body simply don't help keep you healthy. How do cells move? When it comes to cell motility, the majority of the work is done by two organelles, cilia and flagella. Cilia are long, tubular organelles found on the surface of many eukaryotic cells. They have a complex structure and a mechanism allowing them to wave in a circular pattern or snap in a whip-like fashion. So these cilia are small and have a hair-like structures that project out of the cell. So they are driven by motor proteins and they are able to move back and forth in a rowing-like motion by helping to propel the cell forward. Cilia can also move the environment around the cell. For example, the cilia on the cells that line your airways continually row unwanted particles up and out of your lungs. Flagella. Flagella are microscopic hair-like structures involved in the locomotion of a cell. The word flagellum means whip. It has a whip-like appearance that helps to propel a cell through the liquid. Some special flagella are used in few organisms as sensory organs that can sense changes in pH and temperature. So the flagella help the cell moves by spinning around their axis in a corkscrew motion. They move in response to a chemical concentration gradient, indicating a sensory feedback regulation system. This is the basis for bacterial chemotaxis. And also, there is a difference on how their flagella moves in prokaryotic and eukaryotic, or in eukaryotes or prokaryotes. So in prokaryotic flagella run in a rotary movement, or in a uniform rotational motion, while the eukaryotic flagella run in a bending movement. The prokaryotic flagellum uses a rotary motor, and the eukaryotic flagellum uses a complex sliding filament system. The cytoskeleton and the cell movement. While cilia and flagella can drive the cell directly, the cytoskeleton, a set of structural proteins responsible for maintaining the cell's form, also plays a significant role in cell motility. Cells use a protein called actin, a part of the cytoskeleton, to help drive motility. Actin fibers are highly dynamic and they can get shorter or longer according to the cell's needs.
Elongating actin fibers in one direction while retracting them in the other pushes cell forward, allowing the cell to move. So as you can see on the picture, you can see the schematic of the three stages of cell movement. So in the picture, it indicates where the actin network is, the direction of the motion, and the substrate. So in cell, after determining its direction of motion, the cell extends a protrusion in this direction by actin polymerization at the leading edge. It then adheres its leading edge to the surface on which it is moving and de-adheres at the cell body in rear. Finally, it pulls the whole cell body forward by contractile forces generated at the cell body and the rear of the cell. What guides cell locomotion? Chemotaxis is this the response of the cells to chemical stimuli by directed movement is one of the most basic and fascinating processes in nature, involved in both health and disease. Cell motility can also be hardwired into your cells so cells know where to move based on their genetics. Positive chemotaxis, it promotes movement toward a stimulus. Negative chemotaxis means movement away from a stimulus. And chemotaxis, it is the direction of the cellular locomotion is determined by the gradient of substances in the environment. The movement of cells toward a higher concentration of a stimulating substance is defined as positive chemotaxis or attractant, while the movement away is defined as negative chemotaxis or repellent. And this is important because, for example, in the bacteria, chemotaxis was used to find food by swimming toward the highest concentration of food molecules or to flee from poisons. And sa multicellular organisms, chemotaxis is critical to early development. For example, movement of sperm towards the egg during fertilization and development, migration of neurons or lymphocytes, as well as the normal function in health, migration of leukocytes during injury or infection. Types of cell motility. Flagellar motility, like the tails of dolphins and other large animals, they move in such a way as to propel their host cells through liquid movements. And this type of motility is caused by flagella. And depending upon location of flagella, bacteria can swim smoothly, reverse the movement backward or forward or tumble. And amoeboid movement. A cell moves by extending a part of its membrane and cytoplasm and then transferring its cytoplasm into a new appendage. It is essentially a type of crawling whereby the cell pulls itself across a flat surface. And an amoeboid movement it is the most typical mode of locomotion in adherent eukaryotic cells. It is a crawling type of movement accomplished by protrusion of cytoplasm of the cell involving the formation of pseudopodia or false feet and posterior uropods. Warm motility. It is a type of motility practiced by bacterial colonies. When environmental conditions are right, colonies of these single-celled organisms undergo changes to allow them to move across flat surface together. And swarming motility, it is also operationally defined as multicellular flagella-mediated surface migration of bacteria. Swarming requires intercellular interaction, surfactant secretion, and increase in flagellar numbers. Swarming motility has often been genetically bred out of laboratory strain and is best observed in natural isolates. In the laboratory, one must take two standardized swarming conditions, although the specific conditions that promote swarming are species dependent. Swarming generally occurs on nutrient-rich media solidified by agar concentration of greater than 0.3%. Gliding motility. It is a type of translocation used by microorganisms that is independent of propulsive structures such as flagella, pili, and fimbriae. And gliding allows microorganisms to travel along the surface of low aqueous films. The mechanisms of this motility are only partially known. The summary of the lesson. Cell motility is the movement of the cell from one place to another. It's a process that uses energy. Movement is guided by the cell cytoskeleton and can involve specialized organelles like cilia and flagella. Cells can know where and how to move based on genetics. They can also respond to chemical signals from the environment, which is called chemotaxis. Positive chemotaxis is movement toward a stimuli, while negative chemotaxis is movement away from it. Cell motility is important for the overall functioning of an organism. In the human body, it plays an important role in immunity and healing. When cell motility goes wrong, it can contribute to diseases, including cancer.